Hello everyone again. So we're going to welcome the first keynote speaker of the conference. First, a few remarks about the virtual participants. As Marta said, you need to type questions into the Q&A box below the live session screen. And in the audience, if you have a question afterwards, please wait for the microphone to arrive and hold it close when talking. So now it's a great pleasure to invite the first keynote speaker of our conference, María Martinón-Torres. She's the director of CENIEH, the Centro Nacional de Investigación sobre la Evolución Humana, or the National Spanish Center for the Research on Human Evolution, and she is as well honorary professor at UCL. Maria has a PhD in medicine and surgery, and she has specialized, among others, in dental anthropology. She has led and participated in some of the main recent research projects that have contributed fantastic new data to the understanding of human evolution. That includes, for example, her work on Dmanisi in Georgia, in southern China, as well as, of course, on the Atapuerca site with the recent discoveries in the Sima del Elefante. So it's a real pleasure to have today Dr. María Martinón-Torres with us for this first keynote lecture, East meets West, New Insights into Pleistocene Homo Evolution across Eurasia. Thank you very much.
Thank you very much. So, I think the speaker is this one, no? So, well, dear colleagues and dear friends, it is really a pleasure to be here today. It's really exciting, and I, of course, would like to start by thanking the organizers of this meeting for such a wonderful job. Thank you, dear Marta. Thank you, Luis, Alice, Karina. Thank you, Gemma. And this is an amazing organization, a very exciting lineup. So I'm really very grateful for your kind invitation to be part of this. And, well, I'm really looking forward to the coming days and discussions and conferences. What I would like to show you today is like a general overview of the latest hominin findings in Eurasia, with a special focus in the findings from the Atapuerca sites I am very fortunate to work in. And I would like to highlight how these new hominin findings have shaped our understanding of the hominin settlement in Eurasia, but also how these new findings have also brought new questions and challenges in our field. So the Atapuerca sites I work with are, as many of you know, located in Spain, in the northeast of Spain, close to the historical city of Burgos. And given their importance, they were recognized as UNESCO World Heritage Site in 2000. There are many different localities in Atapuerca. Currently, we are excavating up to 10 to 12 different localities at the same time in every season. But among the localities, I will focus today in the ones that I would say that are more famous in the world because they have provided the most relevant human samples. And these are mostly the Sima de los Huesos site, dated to 430,000 years, the Gran Dolina site, which in particular at the TD6 level has provided a human sample dated to 860,000 years. And then we have among the oldest of all of the sequences we have in Atapuerca, we have the Sima del Elefante site that has provided hominin remains dated between 1.2 to 1.4 million years. We will start by focusing in the Gran Dolina site. This is a very famous locality. This is a vertical infilling that has more than 10 to 11 meters of sediments, where we have been able to identify up to 11 different stratigraphic levels. And these sequences range from close to 1 million at the TD1 level, at the bottom, is just above the Jaramillo event, up to 300,000 years at the moment in this level TD10, where we have evidence of a very intense occupation. It was a campsite from the Middle Pleistocene with a very rich lithic assemblage, a nice sample of the fauna remains that were exploited by the humans living at that time, and that gives a lot of information about the Middle Pleistocene populations that lived in Europe. What happened is back in the 90s, the Atapuerca research team decided to excavate an archaeological survey to really explore the potential of the site. And in 1994, they reached the so-called TD6 level. This level was located below the Matuyama-Brunnes paleomagnetic reversal, meaning that everything that is found in this level, TD6, in particular in the one we call Aurora stratum, was older than 780,000 years. And what did we find in that small survey pit, which was no more than 6 square meters? We found an assemblage of more than 100 human remains that were mixed with several fauna remains, and also with about 100 lithic tools assigned to the mode 1 or old one model. This was a big surprise because at that time the prevailing theory was that Europe was not settled by humans earlier than 500,000 years. So this was really pushing back earlier the first date of hominin presence in Europe by far, I would say, a few 100,000 years. Currently, this site has been dated, I would say, by almost all means of methods that are possible and available for a quaternary site, so by biochronology, paleomagnetism, uranium series, ESR, cosmogenics, and thermoluminescence, that allow us to securely date these hominin fossils from the TD6 level of the Gran Dolina to about 860,000 years. At that time, as I said, these fossils were represented the oldest hominin fossils in Europe. So currently, when this excavation pit was finished, the hypodeme of Homo antecessor consists in about 160 specimens that have been assigned to 11 individuals. We have mostly children and immature individuals, but we have up to 2 young adults. And all the skeletal parts are preserved, so it has allowed us in the last decades to really work towards quite of a good morphological characterization of the hominins found at this level. When we start comparing these fossils that were new, unexpected, in the time, in the region where we find with other hominin species, we realize that these fossils from the Gran Dolina TD6 level were displaying a combination of features that we didn't see before in the fossil record. This combination of features was quite unique and consists, on one hand, in the presence of quite primitive traits in their dentition. In a way, this was not a surprise given the age. Okay, we have a hominin that is close to 900,000 years, is primitive, and when we say primitive, we refer to this gorgeous teeth that has like superstructures, has a cingulum in the buccal surface of premolars, these very complex extensive crenulations of the enamel. They have very large teeth that have complicated occlusal morphology because of the additional cusps and ridges. We have like very asymmetric contours for premolars, this very complex and molarized root, all features that, well, are primitive and are in common with other early Pleistocene Homo from any other continent, from Africa and from Asia. However, the surprise was that these primitive teeth came with what we call a modern-like face. And when we talk a modern-like face, a face similar to the one that we Homo sapiens have, we are referring to the infraorbital region, which, as you can see here, in this case, it has a vertical or coronal orientation. It's a face like it's like really flat, even a bit depressed, developing what some people, although it's a trait a bit difficult to describe, have what they call a fossa canina. We could say that these fossils were the earliest evidence of a modern-like face, which indeed is not that modern, so we are really wearing a vintage design because our face was already in the fossil record more than almost close to 1 million years. And this face was indeed showing a departure from the morphologies we could see in other coetaneous populations from Africa and Asia. This unique combination of these primitive teeth together with a modern-like face led to the naming of a new species, Homo antecessor, which was published in 1997 in Science. At that time, based on the expression of several facial and cranial synapomorphies that Homo antecessor was sharing with Neanderthals and with modern humans, these fossils were proposed to represent the best candidate as the last common ancestor of sapiens in Neanderthals. Now we think that yes, Homo antecessor is probably the hominin species that is close to that node of divergence, or at least probably what we have in the Iberian Peninsula is a representation or an expansion into Europe of that population, or the closest we have of how the last common ancestor should look like. This phylogenetic proposal, which was controversial at that time, somehow was sort of confirmed 25 years later with the analysis of the dental proteome. The analysis of the proteins recovered from the enamel of the teeth of Homo antecessor revealed that this population was again at the base of the clade that contains Neanderthals, Homo sapiens, and well, now Denisovans. At that time, when we discovered Homo antecessor, Denisovans did not exist, okay? But now we see that has an ancestral position to the clade that is containing sapiens, Neanderthals, and Denisovans, and again, this means that it could be very close to that node of divergence. Also, at that time, it was proposed that Homo antecessor had an African origin. But these statements were mostly based in the analysis and interpretation of features that are primitive and that were present in other hominin populations from the similar periods in Africa and Asia. Also, I would say that the interpretation of the origin of Homo antecessor as African was also based in a prevailing assumption or general assumption that most, if not all, the Eurasian populations were really having an African origin. So in this context, the general view was that the settlement of Europe was the result of a few high-impact dispersals out of Africa. However, in the last decade, with expanded comparisons of the European fossil record with the Asian one, we think that this narrative has changed. It's almost 20 years since they performed this study that in a way for us was the starting point of our change of the narrative, where we analyzed a wide sample of more than 5,000 human teeth from the Pleistocene times to present times, recovered from all continents. And in this case, it allowed us to identify a Eurasian dental pattern, which is a set of features that characterize the teeth of populations from Europe and from Asia from the late early Pleistocene until the appearance of Neanderthals, and that is different from the African dental pattern, which are the characteristics that are globally shared by the populations found in Africa during the Pleistocene. The possibility of identifying this pattern that maintains or keeps relatively stable throughout the Pleistocene for us was suggesting that the evolution of Asia and Africa was relatively independent for a long period, and that the impact of Asia in the settlement of Europe was stronger than that of Africa. But it's not only about the teeth. I love teeth, but it's not only about the teeth. And I would say that apart from the dental data, in the last years, we have seen that there are other features that are talking about similarities between the Asian populations and the European ones. No? This is a bit the east-west connection. And we have added to the teeth some facial, mandibular, and postcranial similarities, features that are in common between Homo antecessor, for example, and classic Homo erectus from Asia, and that are not present or very rare in their African counterparts. Among these features, we could highlight that, okay, antecessor and Asian Homo erectus are the only populations to present a zygomaxillary tubercle before the late Pleistocene. They have very thick cortical bones, large cranial bases, but I would highlight mostly, as we were talking about, this region, the mediofacial region. What we can see is that fossils like Nanjing, Dali, Chukutien, we also will be talking about the recently published Hualongdong fossils, and even Junxian, have some traits in the mediofacial region that are much more gracile, maybe not exactly like the ones we have in Homo antecessor, but we really have a trend towards gracilization in the face that is very different from the massive prognatic faces we see in the early Homo from Africa. So again, this we interpret as another evidence in line of the connection we have, or the interaction between the populations that are found within the Eurasian continent. This work, this comparison of the European, Asian, and fossil record has led us to propose an alternative model to explain the settlement of Eurasia, and the affinities we found between the Asian and the European limbs. We believe, or we suggest, that after the first out of Africa hominin dispersal, something that we have documented in the fossil record with the Denisovan hominins, the colonization of Europe may have been the result of several hominin migrations into Europe. But then in Asia we would have a similar thing coming from a source population. A source population that is not necessarily located in Africa, but perhaps is located in Southwest Asia, which is a region that has been described as having some environmental and climatic conditions that were relatively stable and would allow for the settlement in sort of permanent basis of a mother population or source population. Evolutionary changes occur in that mother populations and would be reflected in the daughter populations we have in the east and the west of the vast continent. The fact that they share a sort of common mother population would explain the shared features between the European and the Asian populations. These populations in the periphery of the Eurasian continent are not continuously communicated with the mother population, and that gives result to their own speciation and their own features. We also think that probably climatic changes and environmental challenges were the principal driving forces behind this separation and this intermittent pattern of recolonization and probably expansion, contraction, and local extinctions of these groups, and would explain the high variability and diversity we have in these populations. Fortunately, although the fossil record from early Pleistocene is very limited, it seems that we are entering promising periods because we see that the early Pleistocene record keeps increasing. It's already like 30 years after the excavation of the pit in Gran Dolina finished, and last year, almost the last day of the season, we managed to reach again the TD6 level in extension. So, okay, here you see in this image, this is the survey pit, and here we are reaching again all these square meters of TD6 again. So this means that we expect, or at least we hope in the coming seasons, that we may find again more Homo antecessor fossils. So here is just an aperitif. We found this little piece of a maxilla, so it looks quite promising in the coming seasons. But it's not only about Gran Dolina. Fortunately, Atapuerca has provided another site which is relevant for the characterization of the early Pleistocene in Europe, which is the Sima del Elefante site. In this case, this site is also located in the Railway Trench, is only a few meters away from the Gran Dolina, although they are separated by 100,000 of years. And here we have again a vertical infilling where we have been able to identify up to 12 different stratigraphic levels. And what happened is that in 2007, and its TE9 level, we found a human mandible. Okay, by a combination of paleomagnetism, biostratigraphy, and cosmogenic nuclides dating, this level where the human mandible was found was dated between 1.1 and 1.2 million years. This became again the oldest hominin fossil of Europe, by this time 400,000 years earlier than Homo antecessor. And who is this guy? Okay, in the case of Homo antecessor, we have quite a large sample, so it was relatively easier, although it still gives us a bit of a headache to understand Homo antecessor particular pattern of combination of features and how does it fit, no, in the family tree. But in the case of Sima del Elefante, okay, it is a mandible, and it is a fragmentary fossil, so the information we can get is important, but is limited. So what we see when we analyze this mandible, we realize that in the anterior part of the symphysis is primitive. Again, something that is not surprising given its age, close to 1.2 million years. And we can see that has features in common with other coetaneous populations from Africa and from Asia, like in the shape of these hints of amentum osseum, the shape of the incisura submentalis, and the interdigastric spine. So no surprises here. I would say that the surprise comes when we analyze the inner part of the mandible. And here what we see is that it's really very derived in comparison with other coetaneous or similarly aged fossils from Africa and from Asia, that has a subvertical alveolar planum, that has barely an inexistent alveolar prominence, and these features are very derived given its age. So, unfortunately, in the case of Homo antecessor, we only have a hemimandible that is just cut here. So it seems, it hints that this mandible would share that particularity with Homo antecessor. But, you know, we have to be cautious. It's only a fragment of a mandible. So I think that these differences with regard to other coetaneous populations from Africa and Asia may be pointing to a specialization or departure or divergence of the European populations with regard to the rest of the continent. But we have to acknowledge what we have, and we don't think this evidence was enough to make a really sound taxonomic assignment. So in this case, we prefer to simply classify it as Homo sp, Homo species, pending further evidence. And, well, at least we have another document that we even have hominin populations in Europe, again, much earlier than expected. And then Atapuerca strikes again. And in 2022, we were extremely lucky again, and in the same site, in the Sima del Elefante site, but 2 stratigraphic layers below where the mandible was found. So this is TE9, where the mandible we just spoke about, Homo sp, was found, dated to 1.1, 1.2 million years. And this is TE7. A few meters below, we found the partial face of an adult individual, which became, again, in this case, the oldest hominin fossil in Europe, again, for a few 100,000 years. This is a fossil that, of course, brought us a lot of happiness, and I would say, as it happens with science, more questions than answers, but we like that. So, in this case, this fossil corresponds to the left side, most of the midface of the left side of an adult individual, and in this case, we could go a bit farther with what we know or we cannot know. Okay, so we remember with the case of the mandible, we could not confirm or discard if that mandible was belonging to Homo antecessor or not. But in this case, we are preserving a very informative area indeed. Okay, so we have gone a step further in our understanding, and that means that we can say with quite a lot of confidence that 87.1, which has been nicknamed as pink, which is also much easier to say and to talk about pink, does not belong to Homo antecessor. And this is very exciting because it means that we are able of documenting a period of hominin populations in Europe we did not have documented before. Okay? We knew Homo antecessor was in Europe at the late early Pleistocene, but this is a population that is different, so it's an earlier population with regard to Homo antecessor. So we know they are different because, as you can see in this comparison, it really, pink does not show the coronal vertical orientation of the infraorbital plate. Is a much more primitive face, much more massive, prognathic, is inclined forwards, as you would usually find in general terms in Homo erectus sensulato, and also it lacks the projection of the nasal bones. Okay, the projections of the nasal bones is something you find in Homo antecessor and later Homo species like sapiens and Neanderthals, and in this case we could figure that pink, like most Homo erectus sensulato, would really have quite of a depressed and low area in the nasal region. Why don't we include it then in Homo erectus? Again, I think we don't have enough data because when we compare pink with other fossils that we generally classify as Homo erectus sensulato, there is not a perfect match. Okay, here you see a superimposition of the fossil over some fossils like those from the Manisi, some Homo ergaster specimens and Sanjiran. As you can see, is that in general terms, pink has a narrower and a shorter face. So what is the situation? Okay, it could be that this is a Homo erectus, it's a regional variation of a Homo erectus, or could be that it's something completely different, but we don't have the data enough to, again, be too conclusive. So we have decided to name it and classify it for the moment as Homo affinis erectus. Homo affinis erectus is a term that is highlighting the similarities of this fossil with the specimens that we usually classify as Homo erectus sensulato, mostly based in primitive features that are in common to all these groups. But we also acknowledge that the data is not enough to confirm or to discard its attribution to Homo erectus. So could be Homo erectus, maybe more fossils are found and we can really ratify that it's Homo erectus, or it may belong to a completely different population, but we don't know at this moment, okay? But what it is clear, and I think it's interesting for understanding the settlement of Europe, the early settlement of Europe, is that belongs to a population that is different from Homo antecessor. So in that sense, we are really like introducing a new protagonist or a new family member in the story of the evolution of the European continent. And now, also within Atapuerca, we are moving from the early Pleistocene to the middle Pleistocene. And in the middle Pleistocene, I would say that one of the most relevant sites worldwide is Sima de los Huesos. Sima de los Huesos is a very small locality, no more than 6 by 8 square meters, that is very well deep inside a karst system, Cueva Mayor, Cueva del Silo, but in that very small place, we have recovered, I would say, one of the largest hominin samples coming from one single locality in the Pleistocene, in the middle Pleistocene. We are talking about more than 6,500 fossils that have been assigned to a minimum number of 29 individuals. And all the skeletal parts represented. We have males, so we have females, and we have children, young adults, and elderly adults. So it's really giving us a very complete picture of a middle Pleistocene population. In terms of morphology, I think it's a general agreement that this population we found here is very closely related to Neanderthals. We could have debates, which maybe are not the most interesting ones, I would say, at this point about the taxonomy, which name we give to them, but I think we all agree that the population we have found in Sima de los Huesos is very closely related to the origin of Neanderthals. That means that Neanderthals have very deep roots in Europe that really go back to the middle Pleistocene. To put some example of the anatomical parts, of course, I would go with my beloved teeth, and I would say that Sima de los Huesos hominins present all the features in the teeth you would ask a Neanderthal to have. So they really have a fully Neanderthal pattern. But this is, in a way, what happens with the rest of the anatomy of these hominins. The analysis, for example, of the skulls also show that they are really closely affiliated to Neanderthals. It's true that in the cranial they also have some particularities. They also have some more primitive features. For example, the brain size is smaller than what we find in classic Neanderthals. But in general, it's very clear that the hominins from Sima de los Huesos are related to Neanderthals, whether they are early Neanderthals, pre-Neanderthals, Heidelbergensis, a subspecies of Neanderthals will be a different type of taxonomic discussion, but I think the interesting part is that we all agree that they are very closely related to Neanderthals. This is something that, well, was also ratified by the molecular data. You know, Sima has provided the oldest DNA that was recovered in a non-permafrost context, and it has confirmed this very close phylogenetic relationship with Neanderthals. So I would say no surprise here. In a way, the surprise with the genetic data came with the previous analysis, the one of the mitochondrial DNA. What we found is that the closest group to Sima de los Huesos were not Neanderthals, but indeed were Denisovans, a group that, as we know, is geographically and temporally quite distant to Sima de los Huesos hominins. So in these papers, they propose like different scenarios to explain this deep shared ancestry between the middle Pleistocene population from Sima and the late Pleistocene populations of Denisovan in the Altao Mountains. But one way or another, I think that this is interesting because we are seeing again a connection between the East and the West. Okay? And I think these findings somehow are highlighting the weight that the interactions within continents have rather than the interactions between continents, okay? So I really think that the movement and the connections and the interactions between the populations within the continent, in this case in Eurasia, is more intense. But we really have a challenge with Denisovans, no? So what do Denisovans look like? Okay, we know that Denisovans are a sister clade of Neanderthals that have been mostly identified in basis of its genetic analysis. So we have, in a way, its molecular signature, but we almost do not have any clear physical evidence beyond a few teeth and tentatively 2 mandibles. Okay? So if we look at the dental evidence, I would say that the Denisovan teeth are large, are complex, are robust. They present morphologies and dimensions that would be more typically found in classic Homo erectus from Asia. in middle and even early Pleistocene populations from Asia. So they really have a morphology that is very primitive, even also not only in the crown, but also in the root. These teeth, in purity, I would say that these are the only fossils we can say for sure that are Denisovans, okay? Because they come from there, and these are the ones we really have the genetic signature from it. However, and this is very interesting, there are other fossils that have been tentatively attributed to the same group. One of them is this mandible, the CENIEH mandible, which was found in the Tibet. And in 2019, it was attributed to the Denisovan group. Indeed, was presented as the first Denisovan found outside Denisova, based on the analysis of the proteins. I think this is very exciting. I think this is promising, but I also think that we need more data. So I would say, in general, I would be cautious with making taxonomical assignments in the basis of only one amino acid polymorphism. Okay, so I think this is very interesting, and it's not only about the proteins, because in terms of morphology, I agree that this mandible also have very large teeth that are also like bucolingually expanded at the mesial cusp. It seems that they have some features in common, but I think with such an emerging and new field, which is proteomics, we should have a better understanding of the variation of the proteins in wider populations. Along the same line, a few weeks ago, probably you saw it in the news, there was another mandible that, based on the analysis of the proteome, was assigned to Denisova. And it's this fossil, which was published like 10 years ago as the first archaic Homo from Taiwan. Indeed, this mandible, Penghu, that has been fished close to the offshore of Taiwan. And in this case, again, they have it assigned to the Denisova group because it shares one mutation, we say one amino acid that is present in the Tibetan mandible, and another one that in the Denisovan. But again, I think it's promising, I think it's interesting. I would indeed highlight that the Tibet mandible and this one from Denisova share quite of a rare trait in their molar. They have a 3-rooted lower molar, which is quite of an unusual character. They have it in common. They have very thick mandibles that have a low corpus, so there is potential for them to really represent Denisovan. But again, I would say we need to understand better the variation. So to really use proteins for taxonomic assignments, I think perhaps we need more information also on the variability of other Pleistocene populations, because at the end we are talking about single isolated specimens, no? So we don't really have a full understanding of the variability of the proteome in these groups. So what we are seeing now is that the Middle Pleistocene in Asia has a high variability, and that we have to expect that the fossil record in Asia has something else beyond Homo erectus. Probably the fact of finding Denisovan has opened the eyes regarding this, because we see that there are certain groups that we cannot easily classify as Homo erectus, or that we believe or we feel that they have a bit of an unclear taxonomic status. In an attempt to put some order in this chaos, we say, okay, to try to understand what do we have in Asia, let's start from the beginning. We are talking about some fossil samples that we don't have clear whether they belong to Homo erectus, but then we should be clear, are we able of recognizing Homo erectus? Why don't we try to really define what Homo erectus looks like? So this is what we try to do, at least dentally, because to give a lot of information. So in the last years, we have been able of identifying or characterizing a morphological dental pattern that is sharing common to what I think we could define as the classic Homo erectus, no? When we talk about erectus in Asia, and that would include samples like those from Chukutien, Yiyuan, and Hexian. These features are quite characteristic, the ones I'm going to mention, and these are features that so far I have not found outside these groups that we consider classic Homo erectus. And I would say this very characteristic bucolingual expansion of the mesial cusps in upper and lower molars, also in premolars, the expression, as I will show you in other pictures, of some singular derivates in the buccal surface of the teeth, and these very robust column-like roots that do not narrow towards the tip that are very, very typical from Asian Homo erectus, and I would say in particular for Chinese Homo erectus. Other features, and for me is like the most diagnostic, is indeed this very unique folding or crenulation of the enamel dentin junction. Okay, so you see all these little crenulations and wrinkles they have in the occlusal surface at the dentin level of molars and premolars, even in the labial surface of the incisors. This is something quite unique, I would say, that so far I have not found outside these Middle Pleistocene populations from Asia. So could be characteristic or maybe apomorphic or topomorphic of these groups. Also, these populations that we have named classic Homo erectus present other features, which are like the shelf-like protostylid and mesial protoconid. Well, it's these structures here we can see in the buccal surface. And the mesial protoconid ridge, which is also a crest that appears in this cusp. And these features appear quite consistently in all these classic Homo erectus samples, and we don't usually find outside these groups. So, this is interesting in what sense? Because by defining classic Homo erectus, we are able, by default, of identifying a non-erectus Middle Pleistocene group. Okay, so at least we try to put a bit of order. These are the groups that I think that we can classify as Homo erectus, and these others that are missing the features I think we would expect to find in a classic Asian Homo erectus are something else. So now we move to the next step. What are they? Obviously, we all expect that within these non-erectus Middle Pleistocene groups, we have to have those Denisovans, the most wanted Denisovans. So what can we do? The problem is that we really need to match the fecal evidence with the anatomy with the molecular evidence, and it's not that easy because so far we have really only done that with the proteins, which, as I say, I think we need further information and a better understanding of the variability. But we can play or we can speculate, no? So we say, okay, let's look at that group of non-erectus Middle Pleistocene fossils and see what do we think. Okay, this group of unclear taxonomic status have received a lot of names. So now this is the headache of paleoanthropologists to try to understand. These have been called archaic Homo sapiens, Homo heidelbergensis, China archaic, archaic humans, archaic Homo, Homo longi, and very recently, maybe you heard, Homo juluensis. Okay? So these are all those new fossils that we don't have clear where to put, but it seems that we all recognize that they don't easily fit in what usually we call Homo erectus. So are these, or are all these are potential Denisovans or not? Well, until we really get molecular evidence and we can compare them to the real original Denisovan ones, it's a bit difficult to conclude. But we could play a bit with the morphology and say what we would expect. And if you ask me, and this is a bit of, well, say, speculation because we have not recovered the molecular data, among these fossils, I would say there are a few that, if you ask me, would be my favorite to be candidates to represent those Denisovans. And the ones I like more are the ones from Xuyayao in northern China. So this is a sample that in their anterior teeth have some features that we usually would have interpreted as Neanderthal or Neanderthal-like, but in the posterior teeth, they really have, with the shape, with the buccolingual expansion of the mesial cusp and the roots and everything, have some features that are more Asian Homo erectus-like. Okay, so this combination looks like promising, no? So we're seeing a population, Denisovans are a sister clade of Neanderthals, maybe we can expect them to have their own features, but also some Neanderthal traits. So, okay, this could be a potential sample to explore, which, as far as I know, I think they have tried, and unfortunately, they have not been able yet to recover molecular data. Another nice sample would be, in this case, the Xuchang skull. Again, this is in China, in central China. We are talking about a sample that is dated to 100,000 years. Very large cranial capacity is about 1,600 cubic centimeters, and they have been described as having a mixture of Neanderthals and Homo erectus traits. Again, no, this appealing combination of Neanderthal Asian features. But again, we don't know, we are speculating. Or we could also talk about this other sample. This is from the Maba site in southern China that has been described as showing similarity with Neanderthals and the European ancestors. So really have some features that are similar to the Sima de los Huesos hominids we were talking about in Europe. So what we are seeing when we analyze the fossil record in Asia is that we are seeing increasingly more and more features that in the past we were considered that were Neanderthals or exclusive to Neanderthals. But now we have Neanderthal traits in China. What does it mean? Okay, it could mean that we have Neanderthals in China. I wouldn't close the door, but maybe those Neanderthals in China, they have different morphologies because we are talking about very separate evolutionary stories. So we may have Neanderthals, but of course they have time and space enough to diverge to something different. And Neanderthals obviously is a species that we know and we have characterized strongly focused on the European record. But, okay, we may have Neanderthals in Asia, but they are a bit different from what we would expect of an European Neanderthal. It may be that these are the Denisovans, which is a sister clade of Neanderthals. And then Denisovans could be sort of interpreted as the Asian version of a European Neanderthal. Okay, so again, we're seeing more atypical morphologies. Could be that we have Neanderthal traits because maybe those traits are shared with Neanderthals because they are coming from the same source population. When we were talking about the scenario that the East and the West are sharing a common ancestor somewhere, maybe in Southwest Asia, maybe those traits they have in common, they are inheriting them from that mother population. Or maybe it could be interpreting. You know, it's difficult to answer at this point. But what I think is that first we were thinking that Asia was only for Homo erectus. Now we are open to the idea that has to be home for something else, Denisovans. But I would like to add a third problem here, is that maybe we have focused too much in only the identification of Denisovans, but maybe this non-erectus middle Pleistocene group may be holding something else apart from Denisovans, okay? And I would like to talk and to finish this talk indeed by referring to another sample that presents a combination of features that, to my understanding, are different from this Shuchan, the Shuyayao, the Xiahe mandible, okay? And these are samples like those found in the Hualongdong site in central China. This is a sample that has been dated to 300,000 years. As we see, this is a very key period for the diversification of the Middle Pleistocene populations in Asia. It has a low cranial bone, has some primitive features, the cranial capacity just above 1,000 cubic centimeters. They do not have Neanderthal traits, okay? Not in the face, not in the crania, not in the teeth, so in that sense it's different from the rest. Has some primitive features, but have other derived traits that make them different to those that we were suggesting that could be Denisovans, okay? They are very different from the Penhu mandible from Taiwan, they are very different from Xiahe, so somehow they are different. When we analyze the mandible does not have a true chin, but has already like a hint of an area of momentum. And particularly, we were talking about this before, the area, the infraorbital area, it has a completely different orientation. It's also not like the one we have, Homo sapiens, but it's also much more gracile. So it really has a more depressed area. So we are seeing something different, okay? And it's different from the large brain skulls we saw in Shuchan, for example. So somehow, I think this combination of features are suggesting that in these non-erectus middle Pleistocene groups, we may have something different and that may be closer to the Homo sapiens clade. So this phylogenetic space, this proximity to the origin of Homo sapiens, would be similar to the one that has been suggested for another fossil that was recently published, which was the Harbin skull. This Harbin skull has been suggested as a fossil that is very closely related to the Homo sapiens clade. So we could see that Denisovans is a clade close to the Neanderthal lineage, and we may have another population that Ni and colleagues have named as Homo longi, but this could be debatable whether we give a new name or not. But in a way, we see that we may have about 300,000 years ago a population that is more closely related to the Homo sapiens clade. And in this group, other fossils have been included, fossils like Dali, which is a classic work from Professor Bushinshi, and other fossils I would say that I would include, like for example, those from Hualongdong we just talked about. In this case, I would say that if they occupy the same phylogenetic space, I would be more prone if we were to give names, but names may not be the most relevant issue here. I would prefer, in a way, to pay tribute to the work and the suggestions of Professor Bushinski. I would prefer to use the name of Homo daliensis in reference to that phylogenetic space. Somehow he was already suggesting that these populations may occupy. But what I think is interesting is that fossils like Dali, Harbin, or Hualendong may be representing in Asia a parallel to what fossils like Jebel Hirud or maybe Florisbad represent in Africa. So that means that we may have in Asia, 300,000 years ago already, some morphologies of a population that predates the origin of Homo sapiens and is closer to it. So this is an idea that I think we need to explore, that within that variability we may have something else, and of course this implies complex biogeographical scenarios, but we have to take into account that the diversity is much higher than we thought at the moment. Finally, I would like to finish, and I think this is the perfect place to present our new Paleoproteomics and Ancient DNA research group and lab at the CENIEH. We are really launching now a new line. We are working now in equipping this laboratory, which hopefully should be ready and functioning at the end of this year, perhaps beginning of next year. We have strategically wanted to start by working on proteomics. We have already, slowly, a team, which is Samantha Brown, you may know. She has been working in the THUMS, the proteins for the identification of the Denisovan specimens. We have been also developing a research team in collaboration with the Instituto de Biología Evolutiva with Thomas Marques Bonnet, so we are also working with them in the analysis of short-term proteomics. We also have an association with the University of Burgos, so we are also working with them in ancient DNA. So, well, that would be the nice way of jumping, from fossils to genomes, and we're looking forward to also contribute to this exciting field. And that's from my part, that's enough. I'm very welcome for any question.
Thank you very much, Maria. Do you hear me? Yes. Thank you for this wonderful overview. I think it was a perfect introduction to that meeting as well, because it touches many aspects that will be developed in the thematic sessions in the coming days. Do you have any questions in the audience? Yes, please.
Oh, go on. Okay, there's a question online. Aishwarya asks, how can we integrate, this is special for you, Maria, how can we integrate fossil morphology with genetic data when the DNA is unavailable for some specimens, even if we don't get any similar referring specimens to compare?
Well, I think we can go as far as we can go. And if we don't have the data, the only thing we can do is speculate. I think that proteins are very promising because of the preservation issues. We can really go farther in time. So for those periods where we cannot reach with DNA, proteins are giving us an opportunity of exploring relationships. But I insist that I think that proteins are a complete new world. Proteins are also function, so I think we should not put too much weight in the taxonomic or phylogenetic interpretation, but mostly taxonomic interpretation of the protein variation, and that before doing that we have to have a better understanding of this variability of these populations. So I think, well, there was a time that thinking of recovering DNA from these populations sound like science fiction, and it occurred. Then proteins came, it occurred, so I would be optimistic. But if we don't have, it's very difficult to do that connection. I cannot think of other way.
Yes. Johannes?
Hi, Maria. Thanks for the great overview. So what I was wondering about, you haven't talked too much about
Give your name.
Oh, sorry, Johannes Krause from the Max Planck Institute.
We know, but not everybody.
So I was wondering, you didn't talk too much about the hybridization evidence that we have in genomics between Neanderthals and some early say Homo sapiens-like lineage, because the mitochondrial DNA that we find in Neanderthals, at least in late Neanderthals, as well as the Y chromosome that we have in late Neanderthals, has a divergence time which is much too shallow for the overall divergence time of the nuclear DNA that we have found between Neanderthals, Denisovans, and modern humans. So there was definitely some gene flow happening between some African lineage and this whatever Eurasian lineage that you have mostly talked about today, which must be after 400,000 years, if Sima is rightly dated to 420. Some people say it's maybe 350,000 years. Sima, it's not really clear if it's really 420, but something that happened after, because Sima is still looking like this old lineage that we have in the mitochondrial and the Y chromosome lineage, and then it has to be kind of 270,000 years or older because of the divergences that start to happen on the Y chromosome and on the mitochondrial lineage. So there is some gene flow between Africa and Eurasia between 250,000 and 400,000 years ago, based on the genomic data that we currently have. So what some people are now kind of thinking about, and I just wonder what your opinion about that is maybe we shouldn't focus so much on kind of Denisovans and how strange they might be, but more about Neanderthals. And we are often measuring things about Neanderthal-derived states. You also said they have Neanderthal features or not have Neanderthal features. But Neanderthals are, which we know already, a hybridization product. They are actually part of a African lineage and they're part of a Eurasian lineage. We don't know exactly how much that is, if that's 5%, 10%, some people now even say 50 or 60% of Neanderthal is actually kind of mixed between the Eurasian lineage, which is the Denisovan-like lineage, and one is the African lineage. So what I'm wondering is this kind of mixture of traits that you find, where you say this is really hard to understand because you have some derived, some primitive traits here. Could it just be that what's really strange is the Neanderthal lineage itself, because it's a product of two lineages meeting, say 400,000 years ago. So therefore you have this archaic Eurasian lineage, if you want, which Denisovans are part of it, as we know them now. And then you have basically this African Homo sapiens, like eventually leads to Homo sapiens, but much later, but that must have been present there for whatever, 500 to 800,000 years at least, maybe even more. So eventually those two lineages meet. The African lineage meets the Eurasian lineage, and that gives rise to Neanderthals. And then Neanderthals, really, because we're always using it to then compare it to, because we have so much evidence from Neanderthals, we have so much fossils, so we always compare it to, but maybe the Neanderthals are really the weird lineage, if you know what I mean. And actually the Eurasian lineage would be best presented by Denisovans, even though the problem with Denisovans, of course, is we don't really have good fossils now. I mean, there's some really cool data coming up now, which some people might have seen already, so I think we will have some more knowledge how they actually look like. But could it be that this mixture of traits is really kind of the problem of comparing it to Neanderthals, which themselves are not a, if you want, at least genetically pure lineage, because we do know now they're actually a hybridized lineage between an old African and an old Eurasian lineage.
Well, I completely agree. It's very complex, and there are different scenarios that could explain this pattern. But I would say that for me the problem is, as you said, that we know a lot about Neanderthals, but for example, I find a big gap in terms of molecular evidence that we know very little about early Homo sapiens. And for me, that would be the key to even really understand that integration and everything. What do we have of DNA from these early Homo sapiens? We even have Sima, it's 400,000 years, and we even know how these pre-Neanderthal populations were. But do we have any DNA from any early really Homo sapiens, 100,000 Capsae, or even Jebel Irut? For me, that is the key to really be more clear or more safe of how to interpret this pattern. Okay, so yes, there is integration, but even in this case, where we're talking about the mitochondrial DNA, I know that one of the scenarios has been proposed is that there is an African population that replaced this whole. But again, I find that for me, it's not really parsimonious. For me, it's easier to think in a scenario that they already have a shared ancestry and they are sharing these traits than thinking that it's just one population that is integrating this whole, for the whole Neanderthal pool, a new sequence. So what I think is that I completely agree that we need to leave open that Neanderthals have been very useful, and then maybe there is a bias because we have Neanderthals very well defined, but the problem is that I think we lack comparatively same amount of information regarding early Homo sapiens. And perhaps if we really get DNA, we will see those sequences that we consider integrase, maybe they are primitive. I don't know. I just say that even morphologically, when you look at early Homo sapiens, they're very special too. They are not classic contemporary Homo sapiens. They have what we call primitive features, but really understand the polarity, I don't know. So I would say that the key to really go in one or another direction would be to really get DNA from early Homo sapiens, from this pre-sapiens group, from these 300,000 fossils in Africa, for example, or even the 100,000s. I would like to see Capsea, I would like to see other Chinese samples. So I agree with you. It's like the scenarios are open, but I think we are lacking data on the pre-sapiens groups to have a better understanding of that. So I don't know.
I mean, at least for Homo sapiens, we do have modern genomes, right? So we actually have a pretty deep divergence of the modern diversity that we have today, and we can also calculate the population size.
But I would like the real DNA, not the estimation. You know, I would really want to have the DNA from those 300.
That's real DNA. That's DNA of the people today.
Yeah,
it's just
but from the fossil sample. So the ones we're seeing in Asia, I was talking about, if we really have DNA, it would make a difference. Or the ones like Jebeli root, but if we get DNA from there. Or Jebeli root, I think would be a very interesting one. So I don't know, I will leave it open. I prefer to say what I know, as far as I can go, and leave it open, than to really being very close about one scenario versus the other. Yeah.
Thank you very much. I think we can continue that discussion in the social breaks. Thank you very much. We're going to have, thank you very much again.